Hi, everyone. Today we're going to work through an exercise where we use tools to do phylogeny. We're going to use DNA evidence as well as morphological evidence and build a tree and map some characters. This is kind of a stripped down, simplified version of what people would do professionally with a lot more computational power, a lot more, I don't know, a lot more effort. We're going to do the version that can get us through in less than an hour, I hope. So let's see what's going to happen here. So we have the document that I'm going to make available to you, the exercise here. I'm just going to leave it up in the Google Docs. And we'll kind of work our way through this. I also have a little bit of a presentation file I'll start us off with. So just really quickly, we've seen phylogenies. You might have had a lab previously in a course where you constructed a phylogeny. And as I've mentioned a few times in this class, and really just in biology in general, we use phylogenies a lot more to understand what's going on with living organisms. It's also shown up in viruses. So as people are studying the coronavirus these days, you can build a phylogeny of the coronavirus and locate sometimes places where new variants originated, or when this was first getting started, they were using phylogenies to find the closest wild versions of viruses that are similar to this coronavirus that showed up. So lots of really important conclusions we can draw using a phylogeny as our basis, and getting these phylogenies is really a field of study all unto itself. But much more common these days. It's easier to get DNA sequence data and fairly easy to run these analyses. So it's getting easier, quicker, cheaper, and more common every day, really. So this is an example of the phylogeny of flowering plants, showing the relationships among orders. And this is part of a study that also dug deeper into smaller taxonomic groups. So within each order would be families and so on. And all these relationships, really widely accepted now, these relationships are built using DNA sequence evidence. So we'll kind of give you a feel for how that happens during this lab here. So when people get DNA sequence data, there's a whole lot of data out there. And some studies go so far as to try and compare all of it. So we're talking millions and sometimes billions of nucleotides of data. And you could compare all of those in the methods that we're going to show you today. I'll build this up for you from something that's a little more traditional. So before we had the current ability to get so much sequence data, people would basically agree on targeting the same genes across a lot of different groups. And so these are some examples of commonly used gene regions. And I want to point out just a little bit about them. So the boxes, we have 3 boxes separating 3 gene regions here. And the first one here I'll talk about is called MATK. And it's not, from a phylogeny standpoint, it's not terribly important what this gene does. It happens to be located in the chloroplast genome, and it consists of genes that code for proteins and tRNAs, and then also spacers in between them. And so these, this block here, this is the MATK protein. This is a bit of the tRNA, tRNK. This is a bit of the tRNA, tRNK. And then these lines here that aren't boxes, these are the spacers in between. And the functions are a little bit important because regions that have a biological function, those tend not to change over a lot of evolutionary times. They don't change as much because a change in the sequence that determines a protein could be a change that doesn't allow that protein to be functional anymore. So if you make a mistake or an error or a change to the functioning version of a protein, you might get a non-functional protein, and through natural selection, that becomes an organism that might not succeed and might not pass on its genetic information. The spacers, on the other hand, these tend to be more variable because they're a little bit less important overall to the function of the organism, and changes in the DNA sequence of spacers doesn't really have as big of an impact on the function and health of the organism. So we kind of like a mix of the two of these, areas that don't change much over big evolutionary time and areas that change a little bit faster. The areas that don't change much, these allow us to make bigger broad scale comparisons, say between a monocot and a eudicot, and also, across flowering plants and seed plants, non-flowering seed plants. And then these other areas that are a little more variable, you might compare a couple of closely related species using the DNA sequence in that location. What's also shown on here, these are these little triangles here represent primers. This is part of the process of doing PCR, where you would amplify a piece of DNA to get a sequence that you can work with. We're not doing any of that, but I'm happy to tell you about it if you want to get involved. And I also do that kind of research in my lab, so you're welcome to ask me about doing that sometime. So MATK, other regions here, this is a group of spacers associated with some more tRNA genes in the chloroplast genome. So tRNA, FL, these are abbreviations for the amino acids they're involved with. And this is mostly spacers, and the primers are located inside of these more conserved, or in other words, less variable pieces of DNA. Finally, this one down at the bottom, this is called the ITS region. ITS is internal transcribed spacer. And it's part of a region that exists in the nuclear DNA, and it exists as repeated copies. So the benefit of having extra copies is that if you're trying to get this out of a DNA extraction, you get more versions of this. You get better bang for your DNA extraction. So that's kind of why this became favored. It's also in the nucleus, so you get an independent perspective on evolution. You can look at the chloroplast separate from the nucleus and see if they're telling you the same thing. Sometimes they do, sometimes they don't. So internal transcribed spacer, it's in between the ribosomal RNA genes, so 18S, 5.8S, 28S. These are sizes of ribosomal RNA that get made from the DNA in this region. And the spacers in between them, again, they're just spacers. They have a little bit of biological function, but they do accumulate a lot of differences as well. So kind of a mix of not variable and more variable. So we'll see that as we line up our sequences. This is an example of a sequence alignment. This is viewed in the program Mesquite that we're going to look at today. And basically, you would get these DNA sequences by going through the process of amplifying and then sequencing a specific target region that you know what you're going to get ahead of time, or you expect to get it, and you try to get the same DNA region for all individuals. And the differences and similarities are important for making these comparisons. So similarities, we need lots of similarities in here to build our confidence that we have aligned these sequences properly. So as we go across, there are some differences. There are some locations that don't have any differences. So this one all the way across, top to bottom, is a T. This one, top to bottom, is a T. Top to bottom is a T. Here's top to bottom is a C, and so on. Top to bottom is an A. This pattern of having similar sequence across a lot of different species gives us confidence that we've aligned things correctly. Now, the differences are also important, because those differences, if two species share the same difference, they're both the same to each other, but different from other species in the group, that might be a signal that they're closely related. And we basically, in this whole analysis, we're going to take all of the similarities and differences across all the species and try to build a tree, a phylogeny, that has the most consistency with the relationship shown and the similarity among the sequences. So these are different species. Going down here, the ones that have the same symbols are the same species, and then they have numbers tacked on. And so sometimes we get multiple individuals of the same species. You can have variation within a species, variation across species, and so on. So there's really a lot going on behind the scenes when we figure out one of these molecular phylogenies that we're going to build today. And of course, there are lots of publications that result from doing phylogenetic analysis. So this is one such publication. This used to be a little more exceptional. Now it's just totally widespread for people to be investigating species relationships using DNA evidence. But this is one such study. This is looking at a group of pondweeds in the genus Potamogeton. And they are using this TRNT to TRNF spacer region, one of the ones that we looked at before. So they've got the relationships among species. Here's some multiples of the same species. So Potamogeton compressus, several individuals here coming out as closely related to each other. Here's a group of Hohenmagetan Gramineus closely related to each other, and so on all throughout this phylogeny. So when people do a phylogenetic study, the DNA sequences that they get, they should end up in this database called GenBank, or some people say GenBank. And they get an official accession number, so 2 letters and 6 numbers. And that's a unique identifier for a DNA sequence that is described as coming from some organism and some part of its genome. So down here we have a species called Alisma subcordatum. Here's its taxonomic categorization. And then down here it says it's the tRNL gene is part of this sequence here. And it's not very long in this example. So nucleotide letters for that gene. And the benefit of having these publicly available is that you can get new sequences for any species and compare them to what's already been published. So this opens up a possibility. One of the possibilities is that you can get a DNA sequence for an unknown organism and compare it and maybe even identify the organism based on its DNA sequence. And that's more and more a feasible option these days. So we're going to do a little exercise today working on an interesting group of flowering plants. This is a family of plants called the duckweeds, and they include the world's smallest flowering plants. So shown here on this image, this is somebody's fingertips covered in these little green dots, and each of those little green dots is its own plant, magnified here in an electron micrograph. And it actually is a flowering plant. So this is it in the stage of flowering with a pistil and anthers. And sure enough, this tiny, tiny thing makes flowers and seeds. So small you probably never would even notice the plant, let alone its flowers. All right, so I'm just going to keep going into the exercise for today. I think it's probably easier for me to just demonstrate the exercise. If you want to follow along, I'll show you the process of getting the software and so on, show you where to look for that. Everything on this you should be able to do on your own, but given that it involves sort of a little bit of a niche computer program, and it doesn't always work for everybody, it requires JavaScript and so on. And sometimes there's a bit of a learning curve to doing the steps here. I'm going to leave it up to you if you want to do this for yourself, or if you just want to watch me do it on the video. All right, so what we have here in the bold there, it says we're going to open a file. Maybe I'll first point you to the program we're going to use. This is called Mesquite. And if you do a web search on Mesquite project, you should get it. So mesquiteproject.org. So a modular system for evolutionary analysis. It's a very capable program, has lots of different abilities, and we're going to use just a few of those. I quite often just use it for the DNA sequence alignment, but it does a lot more. And it's got a little bit of a trail sometimes to find the download. So here we got download. I'm on a Mac here. You could also do the Windows version. And both of these send you over to GitHub, which is holding on to the published version of these. And then down here, you can download the version that matches your computer. And then as it says, you're going to need to use JavaScript to run this. Some people have trouble getting it to run, so that's why I don't really push too hard for making it work. But I can get it to work pretty well on Mac computers. And hopefully you can too if you're interested in. So I downloaded that, I've installed it. It's down here in my doc called Mesquite. And I'm just going to take the file that's mentioned here, Lemnaceae, so that's the duckweed family, MatK, RBCL, and morphology. I'm just going to drag that onto the program that's already open. And what I've got here is a set of species abbreviated, so it's 4 letters for the genus and 4 letters for the epithet. So land punk is Landoltia punctata. And you can see I've kind of curated this so it's a little bit crisper. But you can see that we have sequences that line up very well in the beginning, and then something goes wacky, right? So something is different here about these sequences, this block of sequences relative to the other sequences. And it's not just a nucleotide difference, there's a length difference here. And if you look carefully, you can see this tracks back to this set of A's here. So the A nucleotide In the top block, there are 1, 2 A's and then a G. In the bottom block, there are 1, 2, 3 A's and then a G. And there are a few ways you could resolve this. You can do manual alignments. And I'll show you just a couple of those on here. What I'm going to do for this is create a gap. I'm going to add a character. And I'm going to use this block moving tool where I can select those guys as a block and pull them over. So doing that makes everything line up a lot nicely again, a lot nicer. And we can see going through lots of similarity, some interesting differences. And usually it's not too far until you get to the point where it's probably not as effective to do this manually. I've certainly done a lot of manual sequence alignment in my day. If you're working with lots of data, you tend to need to do this in a way that's a little bit faster. Let's just maybe try to do a little bit of this. I'm going to create myself a little bit of a block there. And I've done this a lot. It's almost a hobby at this point. But I just want to maybe show you how these things can fall into place if you get them to align just right. So at least in this block of letters, now we've got a block that's looking pretty similar. This area where I push things around a little bit, looks like a couple of these have just a lot of extra A's there. So all of these differences are the result of differences, let's say errors in copying DNA, but sometimes over thousands, millions of years. And so it's hard to, you don't want to like track back to exactly where a change was made. But maybe just keep track that changes are important, and the differences that they create can allow us to tell these species apart. All right, so we're getting, you could go through and fix this. That would take a long time, and things start getting pretty messy. If I look farther along at this cumulative difference, I still see some patterns. So it looks like this block of A's, or sorry, T's is pretty similar across a lot of those sequences. I could probably get this to align okay. One feature I like, picking this up a little bit, is you can zoom way out. There's a feature called the bird's eye view. You can get that here or on the display menu. I'm just going to click on the little bird, and that zooms us way out. And we can see that we started with some good alignment, these bands of the same color. But then we get into the point where kind of a cute little artistic pattern emerging. There's a way to save some time on this. I'm going to align a set of sequences to a reference sequence. So I'm arbitrarily going to choose the first species as a reference sequence. It's also in its own genus, so it might be the most, I don't know, the most neutral comparison point for the other species. And fortunately, it's pretty quick at this. So hopefully after this, we'll get a pretty good alignment. Okay. So not too shabby. Looks like things are aligning pretty well, if not perfectly. So the alignment, when we say alignment, we've created gaps where they need to be. The program created those gaps. And also if there's any shifting that needed to happen, that happened as well. Perfect. Wow, piece of cake. So across, this is a plant family. Some people call it a subfamily, but I'm going to stick with plant family here. We can see some regions where things are almost identical across all these species, right? So lots of identity in this region, a little bit more variation here, especially the gaps that exist in this region. But overall, really good confidence that we've aligned things correctly, and that's one of the most important things. So if you get the alignment wrong, then your tree that you build using your alignment is not quite as meaningful. All right, so maybe I'll pull this over so we can kind of see the exercise and also work on it at the same time. So that was aligning. So I mentioned the manual alignment tool here, and then also the automated alignment tool. And we've got a pretty good alignment there. So it says, make sure your sequences are aligned to your satisfaction before proceeding. And I say, sounds great. All right, now we're going to use this information to build a tree. So in Mesquite, you can go to the Taxa and Trees menu, and we're going to do a new tree window with tree to edit by hand. So this is going to generate a phylogeny, and this is just a default sort of symmetric phylogeny. It's not meaningful because we haven't used any of our data to build it. It's just a starting point phylogeny. So one of the ways you might tell that you've got a good alignment or good phylogeny is that species belonging to the same genus are closely related, monophyletic, so closely related to each other and not to anybody outside of the genus. So here's a couple examples: spear into spear poly. These should be, if they're in a monophyletic genus, they should be closely related, but they are turning out to be in groups that include genus Lemna, genus Wolfiella. See if we can fix that. So we can search for a better tree using this tool. And there's a few different ways to search. We just go with the defaults. So this is basically a computer is going to take the tree apart and put different relationships together. And as it builds a new relationship, it's going to check it against the sequence data and say, is that relationship more consistent with the alignment? Or does that require us to think about things in a way that's not as parsimonious, not as easy? So I'm just going to do all the defaults here. So it's going to try to maximize our tree length. So tree length is basically the consistency, how consistently the sequence alignment agrees with our tree. And we're going to do it with our DNA data. I didn't mention the sequence data we were looking at before are a combination. So it's just a concatenated combination of two genes, MATK region and RBCL region. And this process sometimes takes a while. It looks like we're going at a really good pace right now, so maybe I'll let it go for a little while and describe it while it's working. So it's trying out different alignments different topologies, different relationships among species. And whenever it finds a relationship that's more consistent with the data, then it shifts and accepts that relationship. And so it starts building this up into a tree that makes a little more sense, hopefully. And sometimes you can cheat a little bit and help it along. So maybe we'll do that. All right, so I'm going to stop it where it was, and I'm going to do a little bit of cheating. First, I'm going to establish the root here. So on my instructions, I say that Spirodella is the most distantly related genus. So I'm going to use my rooting tool here, and I'm going to find Spirodella. There's one, and it hasn't matched up with the other Spirodella just yet. So I'm going to cheat and grab those together. So other Spirodella, I'm going to grab it and put it on the branch with the first Spirodella. And now we have a rooted tree, and that should maybe clean things up with these other relationships. So wrong thing. If we're looking at a smaller number of sequences, things go a little bit faster. So I didn't decrease the number of sequences terribly fast right there. Why don't we try, looks like we got most of our lemnas together, and then we've got Wolfia and Wolfiella here kind of interspersed. So we can search within those. Trying to do this in a way that gets us to a pleasing answer without taking too long. So we've got most of our Wolfias, the ones that start wolf, are coming out together, and then our Wolfiellas are coming out together. And I don't think I'm going to have patience to make this run all the way to completion. Especially when it kind of stops. It's working, but you don't see any improvements for a while. It gets a little impatient, right? Let me do one more just in the, whoops, stop. That one always throws me. Do you want to stop stopping or continue stopping? I don't know. Which one are you asking? All right. So we've already got it rooted. I'm searching within the rooted tree. Ooh, it stopped. It must be happy. I talked long enough. All right, so we're going to keep searching and then try to look at these questions here. Did the genera end up being monophyletic? So we forced Spirodella to be monophyletic. Landoltia is only one species, so that's got to be monophyletic. Now we've got Lemna, currently monophyletic. Wolfiella, this is all Wolfiella here, monophyletic. Wolfia, so sorry, they're very similar. Wolfia and Wolfiella. This is Wolfia, is not monophyletic, and that's one that's actually kind of a persistent problem for us as we study this group. So Wolfia brasiliensis is maybe not going to come up as being in the same group as the other Wolfias. One thing we can do, where's my paintbrush? There we go. You can pick a color and paint a genus if you wanted to. All right, I painted a branch. All right, clearly I don't do enough of this. I think there's a faster way to paint all the branches, but if you highlight a group and then keep searching, you might notice that the relationships shake out in an interesting way. All right, question 2. Do your results support the hypothesis that Lemna valdiviana and Lemna youngensis are most closely related to each other? So lemon vald and lemon young, not yet. Looks like a very impatient person here. So I gave up on my lemna, but maybe they'll shake out. Nope. All right, so here's Lemna youngensis, Lemna valdiviana. They are closely related, but they are not so closely related that they are more closely related than either is to Lemna minuta, lemon minu. So maybe something to check into there. So they're not quite each other's closest relatives. Is this consistent with the sequence alignment? And to answer that question, you might go back to your sequence alignment, and you could simply look at the alignment and try to make it make sense. Sometimes you can get like a block of data that go along with a genus. Here's a nice piece here. So this sequence difference here is consistent with this. I don't know if you can see this quite as well as I can. There's a sequence difference here where there's a red color and everything else is yellow. Here's a sequence difference with some different colors. And if I look in that, that's the genus Wolfiella. So from willow cod to willow welwa, there's some similarities within that group and differences to everything outside the group. So that's helpful. You can make smaller scale comparisons. So if I line up these bits that have this little insertion here relative to everything else, maybe that's important. Here's another one. So remember, we were looking at on the tree, Lemna minu, vald, young, similar, closely related to each other, and have some sequence features in common. So closely related here, this pair is Lemna equinoctialis and purpusula, and they're probably closely related on this tree. Yep, equa and perp. So again, it takes some comfort being able to look at this and make these comparisons, but they are consistent. So the tree is consistent with the sequence similarities. What I'm not showing you in this exercise is you could actually get a level of sort of statistical confidence about how good each relationship is, how well supported that relationship is. We don't really have time to go into that just yet. What we will do is use our tree here and try to put some morphology data on there. So we extracted a whole bunch of data. Let me color that in. We extracted some data about the morphology of these different species and built a matrix about them. And sometimes if you're just building a molecular phylogeny for the first time, you might have these groups that are well defined on the basis of morphology, of what these plants look like. And so we have kind of these big blocks again. So Wolfiella has some features that are in common. This number 3 right here is not unique, but it's common across all the Wolfiellas. Having the 0 at this state, having a 1 at this state, there's a unique one. So a few differences that are common, sometimes unique within a genus. And so we can look at these traits, and what you could do is you could build a tree using the morphology data. That would be like a hypothesis based on your morphology data. Or you can do what we're going to do, and we're going to plot our morphological data onto the molecular tree and see if we see any interesting patterns emerge from that. So we have them described as a character character across the top, and then these numbers are character states, which I think are defined somewhere. Let's find out. There's a table on the handout that explains what all the numbers mean for these. Okay, so we're going to follow this advice here, trace case character history. So we have a tree, we're going to do analysis, tree, trace character history using parsimony ancestral states. And we're going to use the morphology data onto our tree. And that didn't make it much better. We'll leave it small. So now we have the coloring here. There we go. Is going to color the branches according to how the analysis would tell us those features might have changed over time. So here we've got a feature that's common to a couple of these species. This habit. Let's go check our I already have some doubts here. Habit, tree. Trace character 2, trace character 3. Organization, habit, 1, all right. Will a dent and call? Okay. I'm not sure what it's doing. It appears to have put some different colors. I think those are the colors I already gave it. So let me regret putting colors on here. Remove all color from all branches. There we go. Okay, so everybody has this in common. This is habit, says they're all aquatic. These are all floating plants. Organization, plants modified as fronds. So those are not different in any members of this group, common across all of them. Here we go. Sorry, I got a little hung up on those first 2 steps. Character 3, frond habit. So 0, 1, 2, 3, frond habit, floating or submerged versus submerged. And I don't remember what 3 would be on there. Double check my data sometime. What we see here is a pattern that's not terribly unique at any point on the tree. So whether these plants are on the surface of the water or under the surface of the water, not coming up as terribly useful as, say, a classification tool here. Here's one that's maybe a little more useful, frond shape. And we can see this goes along with all members of the genus Wolfia, which makes it a little tough if we consider this species, Wolfia brasiliensis, not to be monophyletic with the rest. Then it has this feature that's in common with them, but it's not unique anymore according to the relationships we have. So frond shape, character 4, flat, inflated, or globose. So lots of flat. Green is a few inflated here and there, and then globose, those more like rounded versions of these plants found in the genus Wolfia. So not unique to Wolfia, but or not unique and monophyletic, I should say, according to that tree. Here's a few others. Some interesting pattern here is a pattern where a group of closely related species have a feature in common. So Lemna youngensis, Minuta valdiviana have state 4 for character 5. That would make them having 1 frond vein. Very important, right? Maximum number of veins or nerves in the frond. And then pretty good pattern here of phylogeny pattern of number of veins. We got a big group of Wolffia and Wolffiella having none, no veins, and so on. All right, so we could keep going on this. I'll just flip through. We'll see if there's some big patterns. Here's a pattern found in these unique to 3 genera, Lamna, Wolffia, Wolffiella. Not a good pattern on that one. Here's another Wolffia pattern and Wolffiella pattern. That's a pretty nice one here. So 2 closely related species have a morphological feature in common. That's ventral appendage. So characters that would be useful for classification are ones that are maybe unique to one of these groups. So the green genus Lemna here has character state 2 for root number. State 2, so that's 1 root, right? So 1 root here in genus Lemna, no roots in Wolffia, Wolffiella. That's one with a pretty good phylogenetic pattern. And I think I'll probably leave us with that one. So we used molecular data to build the tree. We used our tree to look at the evolution of traits in the genus, in the family. And you could study that and write a paper about it.